Hello, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for DNA Repair. This video covers the various types of DNA damage that you might be asked about on test day, as well as the oncogenes that could cause cancer that result from the damage. You'll also be introduced to the proofreading mechanisms DNA has, as well as the methods that DNA can use to repair itself. By the end of this review, you'll ideally be able to recognize the components of DNA replication that play a role in DNA repair and describe what those roles are. With these goals in mind, you should be able to answer questions like this one about replication enzymes, because you'll have heard all about the replication enzymes involved in damage control. Damaged DNA is by definition mutated because it has been changed from its original sequence. The damage has to be fixed to ensure the healthiest possible daughter cells. There are several types of DNA damage. One type of damage is that caused by radiation, like the effect the sun has on the DNA in skin cells. Anything that shortens a strand of DNA, like phosphodiester bonds breaking in the backbone, causes damage. A third type of damage is base alteration, which changes the identity of the nucleic acids. The final type to worry about on test day is correct base pairing, where pairs other than adenine and thymine and cytosine and guanine try to form. You'll see this again when we get to proofreading. Knowing the types of damage that can occur is important on the MCAT, but it's just as important to know the consequences of damage. One such consequence is the creation of cancer-causing oncogenes. The primary feature of any cancer cell is that they replicate way more than they're supposed to, and they live way longer than they're supposed to because they fail to apoptose, or die on a genetic schedule. Sometimes cancerous cells break off and metastasize, or migrate around the body. They begin replicating again wherever they settle down, which is what causes cancer to spread. Because the cancer cells reproduce far too often and they refuse to die when they're supposed to, the typical mechanisms that control cell growth don't work when oncogenes are created. The reason oncogenes are so likely to cause cancer is that oncogenes create genetic freak versions of the proteins that would normally control the cell cycle. The genes that will ultimately become oncogenes are called proto-oncogenes. You also possess a natural defense against oncogenes. Healthy DNA encodes for tumor suppressor genes called anti-oncogenes. The entire process is that proto-oncogenes turn into oncogenes, which cause cancer unless an anti-oncogene stops it. However, on the MCAT, keep in mind that the new mutation of an oncogene can also be a cause of cancer. However, most of the time, anti-oncogenes are not mutated, and they prevent cancer, and everybody's happy. While DNA polymerase's main job is replicating DNA, it also plays an important role in proofreading DNA. This is where the base pair mistake from the beginning of this video gets corrected. DNA polymerase can tell the difference between parent and daughter strands because parent strands have been in the nucleus longer, so they're more heavily methylated. Incorrectly paired nucleotides have unstable hydrogen bonds, which the DNA polymerase can detect and repair in the daughter strand. On test day, keep in mind that while DNA polymerase can correct errors in the leading strand and in Okazaki fragments, because DNA polymerase is responsible for making those fragments, DNA ligase cannot correct errors when connecting Okazaki fragments together. Because of this, the lagging strand usually has more mistakes. Proofreading happens when DNA polymerase is actively increasing the length of the daughter strand. Typically, a nucleotide is added and a diphosphate molecule, or a pyrophosphate molecule, is released. When DNA polymerase attempts to add an incorrectly matched base pair, it can sense the unstable hydrogen bonds that form. In that instance, it releases the newest incorrect nucleotide as well from the strand. Once the erroneous nucleotide is excised, DNA polymerase continues elongating the daughter strand. DNA polymerase can correct mismatched pairs during the S cycle, but in case it drops the ball, there are two genes responsible for correcting mismatch errors during the second growth phase. MSH2 and MLH1 produce enzymes that find and fix incorrectly paired nucleotides. The previous proofreading mechanism is actually an example of mismatch repair. The wrong base is added to the daughter strand. A doesn't pair with C. Proofreading enzymes catch the mistake and repair it. The enzymes replace the erroneous A with a correct guanine, repairing the daughter strand. Mismatched base pairs aren't the only errors that need to be repaired. Ultraviolet radiation can cause nucleotides to bond with other nucleotides on the same DNA strand, necessitating the removal of a dimer. Thymine dimers are one example of this phenomenon. When two thymines on the same strand bond with each other, they create a bump that messes with the new DNA's ability to do its job. There are 5 steps for cutting the problem nucleotides out. The first is that the error must be detected. Once found, the ends of the dimer are cut by excision endonuclease. The dimer is pulled out and DNA polymerase replaces it with undamaged nucleotides. Finally, DNA ligase comes in to connect the nucleotides to the rest of the DNA strand. After DNA ligase does its thing, the DNA goes back to its pre-damaged, healthy state. The last major type of repair to be concerned about on the MCAT is base excision repair, which occurs when the phosphate and ribose sugar in a nucleotide are fine, so the helix can still form properly, but the nitrogenous base has been damaged. Like nucleotide excision, base excision has 5 steps. The first is the recognition of the damaged base. A glycosylase enzyme finds it and cuts it out, leaving an apurinic or apyrimidinic site. AP endonuclease finds the AP section and removes it. The final 2 steps, which you can probably predict by now, are DNA polymerase filling the gap and DNA ligase closing the gap. Now it's time to test your progress on the learning goals. This question addresses DNA replication and repair. The correct answer is D. Each of these enzymes plays a role in both replication and repair. To recap, proto-oncogenes become oncogenes when DNA has been damaged. This leads to cancer unless anti-oncogenes prevent it. However, when DNA is damaged, DNA polymerase can help repair it, so most of the time, cancer doesn't occur. One of the ways DNA polymerase fixes damaged DNA is through mismatch repair, where it removes erroneous bases. It can also correct smaller mistakes, like nucleotides with structural damage that distort the helix, and mutated bases, which don't distort the helical shape, but they do mess up the genetic code. Thank you so much for watching this Kaplan MCAT Science Review for DNA repair, and good luck on test day!